In this video, I would like to explain what the Stein lab does and why we do what we do. My name is Wolfgang Stein and I run the Crab Lab at Illinois State University. One of our interests is to understand how the nervous system creates and controls rhythmic activity. Rhythmic activity is produced by neurons or groups of neurons that we call central pattern generators, or simply neuronal oscillators. Rhythmic activities drive our most vital behaviors such as breathing, swallowing, chewing, and locomotion, but they're also involved in cognitive functions and learning and memory. To achieve their function, central pattern generators must remain active throughout the lifetime of a human or animal, which means they must be quite robust against perturbations. At the same time, central pattern generators are required to be flexible as they need to respond to sensory influences and changes in internal and external conditions. Many pathological conditions occur due to failures of producing rhythmic activity, and it is of particular interest to understand why central pattern generators sometimes fail to produce activity and how rhythmic activity is controlled. The questions that we ask address issues that are concerned with how rhythmic activity can be maintained without being too flexible and hence to fail to produce behavior, but at the same time not being too rigid in order to avoid inadequate responses to environmental conditions. What are the perturbations that neurons in central pattern generators encounter? There are, for example, external influences that directly affect central pattern generators, such as temperature. In addition, there are sensory inputs that are transmitted from sensory structures like mechanoreceptors and proprioceptors. Other perturbations may arise from intrinsic sources or, in other words, from neurons that innervate the central pattern generators. The question is how central pattern generators maintain flexibility and at the same time show robustness against such perturbations. What are the limits of robustness? When do central pattern generators fail and why do they fail? Do certain conditions such as stress favor failures or shift the limits of robustness? This then leads to the question how central pattern generators are controlled and which neurons are involved in activating and regulating central pattern generators. Central pattern generators are often controlled by descending projection neurons. These are neurons that possess long axons and descend from the central nervous system. We know quite a bit about these neurons, and in fact, we do know that they are sufficient and necessary to activate the central pattern generators. Despite their crucial role in controlling central pattern generators and rhythmic behaviors, we know very little about how these neurons affect robustness and flexibility. We also don't know much about how they respond to sensory input and how their response then determines the activity of the central pattern generators. Our lab works on these important neurons as they are a point of convergence where central nervous system and sensory input come together to enable and regulate our most vital behaviors.